Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
Okay, so I work for Intel Parallel Computing Lab in Bangalore, and I graduated from IIT Kharagpur in 2005. Then I went to Northwestern for PhD. My PhD was in high performance computational biology, and I'm kind of continuing the same work here at Intel. So without delaying any further, let me just get to the slides. So this work we did. We have been, in fact, this is still continuing. So we have been doing this for about 1.5 to 2 years now, and we have a publication in supercomputing for this particular work, which I'm going to present right now. This was nominated as the best paper in supercomputing. We didn't win it eventually, but it was at least the finalist. So as you can see, there we have a bunch of collaborators. So the first two are from our lab. First is me, and then we have Kiran, and then we have people from IIT Bombay, NUDT China, and Georgia Institute of Technology USA. And Professor Srinivas Aluru is our key collaborator in this, and you can say that he is the lead from outside Intel in this project. So what is the motivation? Okay, the other thing is that we have about 1 hour, right?
Yes.
1 hour for the talk. This talk is about 25 minutes to half an hour. So feel free to ask questions anytime, even along the way. So just to give a background of why we are trying to do this. So our main goal was to study Bayesian networks, but we wanted to have a vehicle to be able to do that. And we found that there is this nice plant, Arabidopsis thaliana, which is considered a model plant organism, and it has about 22,500 genes and 35,000 proteins. In fact, this was declared model plant decades back, and in 2001 NSF launched a 10-year program to discover the functions of all the genes of this plant. And after about $250,000 spent for over 10 years, and some more from other places like European programs, similar European programs, the current status is 40% of the genes still have no known function, and even for the other remaining 60%, at least one function is known. There may be other functions which we are still not aware of. So this the process of finding the function is more of a biological process. So you use a wet lab you have a hypothesis, you basically try to guess a hypothesis that this is the function, then you use wet lab to test that. That is a very long process, and in many cases your hypothesis might be wrong, so it just takes very long. So we thought maybe we can use computer science to help with this problem. So instead of, so one way of doing that could be just suggest more experiments for the wet labs and then collect data through that, and then use some computer science on top of that. But we thought that instead of going that way, let's first see what is already available, what all data is already available. So there's a bunch of experiments which have been done on this particular plant, and there was huge amounts of data available online. So we thought that let's construct a gene network based on that data. And gene network is basically you have a big graph where you have nodes as genes and interaction between genes as the edges.
So this particular one was taken as a model primarily because a lot of experiments are available for the same set of genes?
No, this was declared model long back. So I think the lot of experiments are as a result that it was declared model plant.
Because the experiments are available, and so hopefully the correlations can be found out easily. Is it why it is that?
No, so.
Or is it because of the scale? I'm just curious. Compared with the human genes or a rat gene or whatever it is.
Yeah, so for instance, rat is a model. So similarly for plant, this is a model. So that everybody works on this, and we know that.
Okay.
As I said, it takes maybe one person's 10 years of research to just identify function of one gene. So unless the whole community works.
So yeah, that's why you.
Large amount of experiments available.
Yeah. Yeah.
So what's a gene interaction? You said you just represent.
Edges represent the interaction. So okay, so interactions can be in various ways. So one possibility is that there are in any function, in any biological function, multiple genes work with each other, and when they work, they actually produce a protein which actually does the work. So to give some background of the biology, so our DNA just encodes the information of how the body is supposed to function, but they don't do the work. So the DNA, parts of the DNA which are called genes are converted to or that information is used to make proteins which do the work. So for example, hemoglobin is a protein. So similar things are done. So in many of these functions, multiple genes are involved together. So one possibility is that you just find correlations. What all genes are working together, that is an interaction between the genes. The other possibility is, what happens is sometimes one particular gene, the function of that is just to enable some other gene. So this particular gene creates a protein that enables the chemical process which creates the protein of the next gene. So that's acting as an enabler. So there's a cause and effect relationship there. So the various kinds of interactions are possible. Here we have a correlation based, we have a combinatorial correlation based.
But if there are many different underlying relations, capturing them explicitly in the database may be more informative, right?
That's true. So that's why when you use the mathematical model to model this thing, you go with that goal in mind that you want to capture these kind of interactions. So in our case, we were trying to capture the correlation based ones, which basically means that they are either co-expressed or they are not co-expressed, something of that sort. So there are, like I said, there are various methods, mathematical models are proposed. There's just a Pearson correlation based one. There are graphical Gaussian models are also available. Information theory based models are also there. And then we have Bayesian networks. And what happens is that from top to bottom, the accuracy and applicability of the model increases, but at the same time, the time required to learn that model from the data gradually becomes a bottleneck. It gets bigger and bigger.
So you are trying to learn the network from the microarray data.
Yes.
So when you say that time in automated implementations, or are they used to in a manual fashion?
No, these are all mathematical. So there are computer programs to do all of these. Yes. So for example, Bayesian networks is an NP-complete problem. So it has exponential complexity. So there was a study conducted across all these models, and what was found that many of them do really poor. They perform really poorly, and 1 in 3 do no better than random guessing. So the state of the models is really bad right now, and so it sort of makes a case for a more complex and more accurate model as compared to just doing anything. Okay, so Bayesian networks, as they are among the most accurate models for this so far from what studies say, we decided to use Bayesian networks for this. And what it does is that it outputs, a Bayesian network is a graph, is a directly acyclic graph, where based on a scoring function, you are trying to maximize that particular scoring function for that DAG. So you're trying to find the best DAG which maximizes the scoring function. And I'll come to the scoring function later on. It was found that it has super exponential search space. Number of possible Bayesian networks you have to search through is super exponential, and it is NP hard even for bounded node in degree. So even if you bound the in degree of every node, even then it has and it's NP hard. So there was this nice serial method which was developed, which is a complexity. So if n is the number of genes or number of random variables, then it has exponential complexity in n, and even for n equal to 20, it takes 50 hours. And like I said in the beginning, Arabidopsis has 22,500 genes, so n is that big there, so we definitely cannot use that. There are various other parallel algorithms based on the serial algorithm, but they are also much slower compared to what we need. And then there are various heuristic based algorithms. So one, for example, is what they do is they are creating human gene network. They take these 13,731 genes, randomly create, so 50,000 times they do this. They randomly take 1,000 of these genes, create a network, which is, it's easier to do that for 1001, and then combine them together to create the whole network. But that will never give you the accurate picture, because across these 1000 subsets there might be interactions which you'll miss. So coming to the scoring function. So so as I said, that we want to find the best DAG which maximizes particular scoring function. In this project we focused on decomposable scoring functions, such that for each node, if you can compute this particular function and then add up across all the nodes, that is the scoring function you have. So, and what this does is, this basically says what is the fitness of choosing PAX as parents of X, where PAX is a subset of all the genes, all the other genes. So since it's decomposable, what you have is, suppose this is your Bayesian network. So for example, B has these two parents, E has this parent, and so on. So for each of them, you can compute this scoring function, then sum up over all of them, and that is your final score. Based on this, we came up with this heuristic parallel algorithm. What we do is that we conservatively estimate the candidate parent set. So for each gene X, we estimate this should be the candidate parent set, and we are going to look into only these as possible parents. And and we do it conservatively so as to not miss out on actual parents. Then what we do is, so to do that, we have used a pairwise mutual information based implementation, which was published by these guys. Okay, I'll skip this. So then, after we have this candidate parent set, what we do is we define a threshold T for all the candidate parents for which the size of the candidate parent set is less than T. We directly compute the optimal parent sets from those sets. For the remaining ones where the size is greater than threshold T, we use this particular expression to reduce the size of them based on what you have already found out. And then, even then, if there are some which the candidate parent sets are still bigger than the size T, we just take the top T correlations. So this is where we apply a heuristic here. But beyond this point, what we do is, now we have only candidate parent sets of size T or smaller. We use an exact algorithm to find the best optimal parent sets from those. And then once we have this, there will be some cycles in the graph which we detect and break, because we wanted a DAG. So through this method, what we do is we combine the precision of exact learning with the scalability of a heuristic learning solution. And the idea is to push the limit of T as much as possible using massive parallelism, because the higher the value of T, the more accurate this output would be.
So two questions. One, in reality, genes can have feedback cycles. So is there a reason to assume DAG here? Is it because of computation tractability?
No, actually, so there are two things. So we have two outputs. One is before we remove the cycles, and one is after we do that. The one which is after removing the cycles is a Bayesian network, because we wanted this to have general applicability also. We wanted to output Bayesian networks also, but we have the other solution also, and to biologists we have given the first one with the cycles.
And the other question was, maybe you might talk about it later. So you said you gave some estimates, right? 1 out of 3 are as poor as random guesses or something like that.
Mm-hmm.
So how do you know what is correct and not? So do you have the ground truth?
I'll come to that. We don't know the ground truth per se. Well, for some cases we do, but I'll show you. We have done some experiments to evaluate this method. I'll show you those. Okay, so like I said, we want to use massive parallelism to push the limits of T. So we have used these two machines, Tianhe-2 and Stampede. Tianhe-2 is the topmost supercomputer, the fastest supercomputer in the world right now. It has been there for almost 2 years now, in fact. Stampede is also another one among the top 10. And one other reason of using this is that they have Intel CPUs and Intel coprocessors. So like I said, the earlier solutions were too far away from solving the actual problem. So we have, through our implementation, we have got a speedup of about 200,000x over the earlier solution to be able to do the entire plant. And now we can actually create gene networks of the entire Arabidopsis plant. So the baseline was working on 1024 cores. This was non-vectorized code. What we did was we implemented a new parallel algorithm, and since we were trying to scale it to up to 1.5 million cores, even though we are increasing the number of cores by only 1500, the speedup we get is 5300 because of the new parallel algorithm. On top of that, we have tried to avoid as much as possible. We have tried to avoid redundant computation and got another 6x. There was load imbalance across the nodes, so we have implemented our own dynamic task scheduler that gives another 10%, and 10% might look like a small number, but for a 55 petaflop system it's 5 petaflops, which is bigger than most of the systems in the world. And then on top of that, we have applied vectorization because we wanted to maximize the benefit of the modern architectures, and in total we have 200,000x performance improvement. So to coming back to the problem, so now for each node, what we are basically doing is we have a candidate parent set for each gene, and then we are trying to find the optimal parent set. The exact way of doing that is compute the scoring function for all subsets of CPX and then find the best one. But so that can be represented as nodes of this hypercube, where each node represents one of the subsets. So you just compute all the nodes of this hypercube and you have the final and find the best one, and that is your answer. But maybe there is a better way of doing it. Maybe we can reuse some of the computation. So for example, if you're processing this set 1, 3, maybe the work which we did for this set can be used here, because at least one of them is common. And similarly for 1, 2, 3, maybe we can use computation done in any of these things. So that's why there are these arrows here. And the other thing, so we are doing that. So whenever we reuse the computation on a node from a node in which the difference is only one in the subset size, the difference is only one, and there's only one element which is missing there. The best way of doing that would be a DFS, because that way you will ensure that, for example, if you're processing this, all of these have been done before that, and you can reuse from any of them. But DFS, this hypercube is only of dimension 3, but we have the T which we are using is of size 32, the best T which we could get to. So this hypercube will be of dimension 2 to power 32. There you cannot store all the nodes of one level in the memory. So we could not do BFS, but we have done DFS to avoid getting into that problem, and we don't lose much on performance. So the challenges of, now we have to parallelize this. The challenges are that the available parallelism is limited to the number of genes which we have, and the amount of work which has to be done for each gene, that varies exponentially in the size of candidate parent set. So there's huge variation and there's very little parallelism available. So we need to do something about that. So for example, suppose we have this 4-dimensional hypercube which we have to evaluate for a particular gene. Instead of processing this as one, what we do is we divide this into smaller hypercubes, and we define a threshold R. So any hypercube which is bigger than R is split into R-dimensional sub-hypercubes, and any hypercube which is smaller than R will stay intact. So this gives us a very large number of work items. So each sub-hypercube can be a work item. Now we have to compute all these work items in parallel and as quickly as possible. So then, but before that, let me just mention the benefit of this reuse of computation. So we have conducted this experiment from 128 nodes to 2048 nodes, and we see roughly a speed up of 4.8 to 6.4x in these cases. So there's a huge benefit of reusing the computation. So we would want to do that, but that sort of introduces this dependencies between one compute node to another compute node. So okay, this is fine. So as I said, we have these work items, large number of work items which we have to process together. So what we do is we use dynamic work distribution and load balancing. So instead of on a small system, what we could do is we could have a master slave kind of a system where one node is distributing work to every other node. But since we are working on a system which has about 8,000 nodes in it, that one master node can become bottlenecked. So instead of doing that, what we have is a hierarchical tree structure. So if this blue thing is the initial amount of work which we have, so that is all the work items together, all the sub hypercubes together. Initially, the root node keeps some to itself and allocates part of it to these other nodes, to the child nodes, and then they in turn do the same thing. So all of these nodes, all these nodes which are parent of any node, are keeping some allocation to themselves and allocating the rest to their children. Suppose one of the child gets done with its work, it can request more work, and this node can use the remaining allocation and give some work back to that. If this one runs out, then it goes to its own parent, and so on. So through this dynamic task scheduling, we have got a performance improvement of about 7 to 18% over the static allocation of work.
So, but you use some particular tree structure of this compute nodes, is that?
So this is a software tree structure. It has nothing to do with the actual topology of the network.
Right.
So we just create a software tree network. The fan out I think we were using of 16. So the root node has 16 children, and then they have their own 16 children, and so on. Or
I was wondering why you use that tree structure.
Because the other possibility could be just use one master node and all the other could be slave nodes. But then since we have about 8,000 nodes, one master and 8,000 child nodes, the master node will get bottlenecked. So to avoid that, we have created a hierarchical structure. So the first one has 16 or 32 children, then each of the next layer ones have 16 or 32 children, and so on. So I think with 8,000 nodes, we were done in 3 layers. So we had a 3-layer structure.
How much it keeps for itself and how much it is the work cube?
That is a parameter which you can define while running it. What we found was that I think 0.2, so 20% of you can keep 20% to yourself, and remaining if you can distribute, that works very well for this particular application. But this may be different for application to application, and we have just provided a parameter which you can tune to get that. Okay, so just a little bit more detail on the reuse of computation, how we do that. So suppose you're calculating the scoring function here of a gene X4 considering parents as X1 and X2. So suppose this is the data which you have. So there are these various cases or various experiments which are done by biologists for which you have got the data for these 3 genes. Now different colors represent different values here. So what you want to do is, so one particular key bottleneck step in the score computation is binning of this data based on the equality of the vector. So this one is not included in that binning. So for the first 2, if you see these 2 are equal, then these are all equal. This is separate and this is separate. So that binning process is the key step there, and then based on that binning, some probability is computed using which entropy is estimated to get the final score.
So what's the colors indicate the mutual information? Is that what is that?
The colors indicate different expression values.
Okay.
So think of this as these are just random variables with different values possible. So different colors are just showing those different values of the random variables.
The dark side indicates whatever. And if you look at all the experiments for all the genes, we have very sparse matrix.
No, we have expression values of all the genes for all the experiments which we have included.
Each experiment will give some.
The expression value for all the genes.
Every
Yes.
pair.
Yes. So now suppose we want to compute this new one in which there is one more added here. So that basically is represented in this data. So instead of doing this from scratch, what we can do is we can add this data corresponding to X3 to these existing bins, and then rebin the existing bins into smaller bins. So this process basically what it does is that no matter how large is your set, you only have to do this last step of rebinning what is existing there already. So this is the reason why we get the benefit of reuse. and I won't go into more details of that. Okay, so experimental results. So we have taken the data set from 11,000 experiments, each measuring all the 22,500 genes. Various cleaning methods have been performed on the data set. Then what we did was, so it helps if, so since I said that different subset of genes participate in various functions. So not all the genes are going to participate in all the functions. So it helps to categorize the data or classify the data based on functions or body parts and stuff like that. So we have used two particular classification techniques. One is the tissue type, which tissue in the plant is it, so the whole plant or various, for example, seed and leaf and all that. And the other is experimental conditions. So what biological process is going on. So for example, based on what is the reaction to light, for example. So based on this we have two classifications, and using these two classifications we generate about 100 different sub data sets of this large full data set. And for all of these data sets, we have, for some of these data sets we have created these networks, and this is the overall strong scaling results which we have. So for two of these, the largest data sets which we had, we go from 256 nodes of TNA2 to 8,000 nodes of TNA2, and the straight lines are the represent linear scaling, and as you see the scaling is not that bad. In fact, we achieve about 75% efficiency going from 256 to 8,000 nodes. Also, we did another study to find what is the best possible for our hardware for the entire system. So possible efficiency was this much, and what we measured was about this much. So it's not too bad. The other thing is that we just to show what is the time to solution and the other things related to this one. So there are these 4 data sets, and for all of them we ran them on Stampede. And for example, these two took only 269 and 501 seconds, so we did not bother to run them on TNA2, which is a much bigger machine. So these ones which we ran on TNA2, they finish in 2 minutes and 3 minutes. So the entire data set can be done in just 2 to 3 minutes. This before this result, could not be done in less than 6 months. And the other interesting thing to note is that remember I had this heuristic where only candidate parent sets which are smaller than the threshold are processed in the first iteration and the remaining in the next iteration. So this is just to show how many of them actually fit in the first iteration itself. So for example, in this case I think it's about more than 98%. In this case it's about 90%. So even in the worst case we fit about 90% of the genes in the first iteration itself. So there is no heuristic involved there. So we have accurate results for at least 90%, and in some cases up to 98% of the genes, and for remaining 2% we might have some inaccuracies. So to evaluate this, we studied the photosynthesis process. So on the right side you have the photosynthesis, all the genes which are involved in the photosynthesis process. This is a pathway. And on the left side is what we did was we had this entire network. We took some of the genes which are involved here, and using them as seed genes extracted sub networks of the big network. We deliberately kept some of these genes out of the, we did not use them as seed genes to check whether they appear in the sub networks. So these pink ones, okay, this color is not looking similar to what I had intended, but this is pink. So the pink ones are the seed genes. The green and the blue, so this is blue and this is green. So the green and blue ones were which we deliberately kept out of that, and we found that they also appear there. So we were already aware that they are part of the photosynthesis process and they appear in that. So that is some sort of validation. And then we found some of these yellow genes which also appear there, for which there was no known function before. So we picked 2 of them, and I'm obviously showing you the cases where we got more interesting result. There are other cases where you may not, but we picked these 2. And what we did was we took a normal plant. In the seed we disabled the first gene here and disabled the second gene here, and this is a normal plant, and they are all grown to the same number of days. So we see that the growth is stunted if you disable these 2 genes.
So you mean here you did this experiment?
No, this was done in Georgia Tech, yeah. So Professor Srinivas Aluru knows a few biologists. He's collaborating with them to do these experiments. So to understand that further, that result further, we created these graphs. So the first one is a normal expression level of this gene on the x-axis. The second one is the expression of this gene when the first gene is disabled. The third one is the expression of this gene when the second gene is disabled. So you see that in some cases the second bar are much bigger. So these are the genes for which that gene was acting as a controller, and since that is removed, they start spiking up. Similarly, third bars are somewhere, in some cases the third bars are smaller. So this in the third case, that gene was acting as an enabler, and when you remove that thing goes down. So using this, we can understand that exactly what genes are being affected by those two genes, which we found to be important in the photosynthesis process. So this gives a very good result to the biologist to go and do further experiments and figure out what all can be understood from here. So this is the final result. And then just to conclude, we have contributed to Bayesian networks. This is the first massively parallel Bayesian network structure learning algorithm. This is applicable in various other application areas. Whichever application areas are using the same scoring function, this can directly be applied. Otherwise, the scoring function will have to be re-implemented and the remaining framework stays the same. Contributions to systems biology, we have a full gene network of Arabidopsis thaliana now, and this can serve as a vital resource for future Arabidopsis research. And there are other observations. Some of the things which we have done in this project are very widely applicable. So for example, this dynamic work scheduling algorithm. In fact, we just submitted a paper just on the dynamic work scheduling algorithm, which was initially done for this work, but then later on we improved it further and then submitted a paper recently. And just because I'm part of Intel, I need to show you this performance notice, and we can get back to questions.
So you mentioned this DFS through the Hypercube. How does that interact with the subsequent tree-based dynamic task scheduling?
Okay.
There's a data flowing according to the DFS tree. And there is a dependence. So
yeah, I understand. So some of the things here are a little confusing, and I understand that. So what is the reason? The thing is that the tree which is used for dynamic work scheduling, and the DAG which is produced elsewhere, and this reuse and in which we have created this Hypercube, there are three different data structures. They are not related to each other in any way, but because we have very tree or graph-like structures in all these cases, they seem that there is some connection.
But the DFS tree determines data flow, right?
So
the data computed, it also reflects a dependence.
So yeah, let me just answer that. So this is the DFS. So okay, so what we have is, for each gene, we have a candidate parent set. We want to evaluate the subsets of all of them to get the final best one, right? So
this entire computation runs within a single
gene
machine.
No, this is for a single gene. Okay. So for a single gene, for each gene we have these hypercubes. Now we have to parallelize that across the entire system. Since there are only about 22,000 genes and we have 8,000 nodes, if we just give some genes to each node, that is not going to give us good load balancing. So we want to reduce the size of the work item which we are distributing. So what we do is we take this hypercube of each gene. For the hypercubes which are bigger, we divide that into smaller pieces. So for example, if a particular gene has a so let's say R is 16, and or let's make it easier, let's say R is 3, and we have a hypercubes, one gene has a hypercube of dimension 4, so we will divide that into two hypercubes of dimension 3 each. If one gene has a hypercube of dimension 5, we'll divide it into 4 hypercubes of dimension 3 each. So for some genes we'll have a much larger number of possible hypercubes which we have to compute. For some genes we might have just one if the dimension is less than or equal to 3. So this gives us a huge number of work items which have to be computed. So now we take each of this, we call this one work item, and then we use this distributed scheduling to schedule the work items. So the work item which is scheduled is one hypercube to one node, and that hypercube is evaluated by one thread.
But subsequently, then you have to combine information from different.
Yes. So that is not a bottleneck at all.
On an average, is it known how many genes does a gene interact with? Usually getting to is how dense are these genes?
So generally people think that each gene interacts with 4, 5, or in most cases 6, 7 other genes. That is the general assumed knowledge, but nobody has done that work at the entire genome level. So nobody actually knows what is the reality. So as one thing which we were very interested in knowing is what do we get, and we actually got very small number of genes which are interacting with each one. So that I think the numbers which we got are also 4, 5, sometimes 2, sometimes 1 is what we also got. So that kind of validates that assumption, but that is a much bigger question than what this study can solve. So maybe in a few years we'll find out.
How do you first to discover all the gene functions?
The reason why I was asking this was because, as you said, this gene networks are horribly sparse. You can actually recover the global structure via these sparse recovery techniques. Have you looked into those?
No.
Okay. So again, it depends on what sort of scoring functions you want to use. So it's an NP-hard problem in general, but if your scoring functions are nice, then it is possible to use iterative techniques, which can be, again, they're embarrassingly parallelizable and all. You can recover the global structure of probably you can recover the global structure without having to make ad hoc assumptions about, if you have a DAG, you break the DAG somehow. That's an ad hoc step. It's not principle. But you might want to look into that as well, because that just takes the problem into a totally different domain. It's not related to Bayesian learning or anything at all.
Okay.
It has more to
So
do with compressed sensing and sparse recovery.
So I would like to understand more of that because so far I haven't understood much. And the other thing is we are not breaking any DAGs here. We are just.
Okay, I thought you said that if you did discover cycles, you would break them.
Okay, break to make a DAG. Yes, that's what we'll. Yeah.
So that's what I meant. So in these direct approaches, because you just if the global solution. Okay, the one other, I'm not sure if these methods are capable of discovering DAGs if required to, but they will give you the best possible solution. If the best possible solution has a cycle, I'm not sure what these methods will do, but they will definitely be able to recover, given the gene expression data, the best network that explains that gene expression data. That's what you are trying to do.
Okay, maybe we can meet offline, and if you can give me some pointers to this. Thanks.
So the reason for using Bayesian, like your choice, was motivated by what other people have been doing in the past?
Yes, the common knowledge was that Bayesian networks are most accurate, or among the most accurate.
No, in this field, I mean.
In this field, yeah. But the other thing is that nobody has created a Bayesian network of this scale, so you cannot really say it, but unless you have actually done the experiment.
So the compressed sensor method you are talking of, so here with the Bayesian network you also get the probability values along the edges, right? So that is what, so for instance, I'll give you an example. If you might want to use an heuristic regression framework to predict those probabilities. So the gene expression data is inclined towards predicting, or rather the data is indicating towards a certain kind of interaction between two genes, let's say, and let's say you want to encode that interaction as a probability value, as in, like, if I fire, what's the chance I make me fire, something like that. So these methods will again encode that. It's just that they will require some function, let's say a heuristic regression function, to take something and throw out a probability value at the end. Yes, so they will give you probability values for each of the edges. But the thing is, the nice thing about these methods is that they will directly output a very small number of edges. they'll not require you to discover a large number and then throw away, which many techniques do. So graph recovery has been well studied in machine learning. Yeah, so these techniques are nice because they directly give you the optimal structure and not require to go back and forth.
But to get that optimal structure, you might have to evaluate the possible search space, right?
Okay, so the exact mechanics of the method will of course depend on what is the problem you're trying to solve, what are the scoring functions. But again, there's not just one technique. There are iterative techniques, there are greedy methods, and there are optimization based methods. So you can, for many of these problems, you can convert the entire problem to a huge LP or a QP, and then spend all the effort trying to solve that QP or LP as well as possible. And the better you solve that, the better solution you get here.
Okay.
But these can be very non-scalable. But so there are iterative techniques as well, gradient descent like techniques and greedy methods, which so the greedy methods might resemble the kind of steps you are performing as an estimate a parent text structure and refine it. So greedy methods do something like that, but it's not exactly. Again, the kind of computations you will be requiring are again some of them are local, but then there will be global computations required as well. So I'm not sure whether these global steps can be parallelized as well, but the local steps definitely.
Okay. Any other questions?
So are you planning to apply this method to other model organisms like E. coli or?
Yes, eventually. Currently we are doing a lot of experiments with Arabidopsis itself, or my collaborator.
Evaluating the network.
Yeah. So first, as soon as we have a good enough validation done and we are very sure of these things, then we can start applying to other ones.
There is this worm that people are very interested in. It's called C. elegans.
Yes.
Has anyone tried doing this with C. elegans neural network, the neuron network, whatever? Because it has just 205 or something neurons. So that's very popular in computation neuroscience. Right. People love switching one of these things on and see whether it stops walking or it stops eating.
There's a company which is studying the interactions between these neurons in C. elegans, and they have a graphic interface also. If you want, I can send you the website.
One of my friends got a master's degree out of torturing these.
We have somebody who's doing bad operations.
Okay. Any other questions?
So I have just a trivia fact about a particular plant. I found that from Wikipedia. Earlier in the morning, I was reading a paper on mining trivia from Wikipedia. So it was very interesting that when I heard this talk, I actually manually did the trivia hunting. So this particular plant was believed to have the smallest genome among all flowering plants when this exercise started. But later it turned out that there were some other flowering plants which had even smaller. So that could be the reason, I mean, the belief that it had the smallest genome could be the reason why everybody got into.
Yeah.
Yeah, different things here. Like rat, for instance, is because it is most close to human genome. Then E. coli, I think, is the smallest eukaryote genome. So different, we choose different model organisms based on different.
Yeah. Plus, you cannot torture humans.
Yeah. 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 Very close to chimp is closer, but again, doing all these experiments on chimp is more expensive than on rats.
Yeah.
Rat shares some 95% of the genes or something.
Okay. Thank you.
Thank you.